your slides.
Okay.
Okay. Okay. So, uh,
Yeah.
it's my distinct pleasure to introduce our esteemed speaker today, Professor Frank Stubenrosh, a leading figure in the field of medical virology and papillomavirus research. Professor Frank heads the Papilloma research, Virus Research Group at the Institute of Medical Virology and Epidemiology of Viral Diseases, University Hospital Tübingen. His scientific journey spans several prestigious institutions. Over decades, Professor Stephen Roche has made seminal contributions to our understanding of human papillomavirus biology, particularly in the molecular mechanism governing the viral replication and transcriptional regulation. His research has elucidated the role of viral proteins such as E8, E2C, and their interaction with host cellular factors like NCOS smart repressor complex and SP100 proteins, shedding light on how HPV modulates host response and maintains persistent infections. His work, published in leading journals like PLOS Pathogen and Journal of Virology, has significantly advanced our understanding of HPV pathogenesis and its link to tumorigenesis. Please join me in welcoming Professor Frank, whose lecture today promises to offer a deep insight into the detail of papillomavirus.
Thank you so much, Khalid. I'm not sure whether it's with the deep insights. I'll try to address some of the problems or some of the issues that we know about papillomaviruses. Here you see my outline. I will talk about the clinical relevance of HPV infections, and then I will introduce to you the viral replication cycle, and then I will move from there, trying to explain to you how the viral replication cycle is linked to the HPV carcinogenesis. And finally, I will tell you something about prophylactic vaccination and therapy. And finally, we will close this session with the HPV diagnostics. So first of all, I would like to talk to you about the history of papillomaviruses. Already 2,000 years ago, where's my pointer? Already 2,000 years ago, a Roman doctor became aware of the fact that genital warts were presumably transmitted by sexual contact. And then 2,000 years later, again, a guy from Italy revealed that widows, married women, or prostitutes, but not nuns or virgins, develop cervical cancer, also linking sexual intercourse with the development of cervical cancer. Around 1898, a British group showed that that dog warts can be transmitted to other dogs by filtrates from papillomaviruses, from papillomas, indicating that a virus might be involved. Cheerful, a guy from Italy, demonstrated that when you take human warts and grind them up and infect other people, you can transmit human warts, also providing evidence for the existence of a virus. The first evidence that papillomaviruses have carcinogenic potential came from Rouse et al., where they showed that a rabbit papillomaviruses can induce cancers in animals. And finally, in 1949, Strauss and colleagues demonstrated by electron microscopy that there are viral particles in human warts. And then Harald Zurhausen in 1974 hypothesized that cervical cancer may result from infections with the virus that is found in genital warts. In the late 70s and early 80s, when large-scale molecular cloning started, it was revealed that there's not one papillomavirus, but that there are many, and they were cloned. And finally, in 83, Matthias Durst, when he was in Zurhausen's lab, cloned HPV-16. In 95, the WHO recognized that HPV 16 and 18 are carcinogens for humans. And finally, in 08, Harald Zuhausen received for formulating this hypothesis the Nobel Prize for Medicine. Papillomaviruses are one of the largest virus families that we know that can infect humans. As of Friday last week, the PAVE database, which I highly recommend for information about papillomaviruses, annotated 399 animal papillomaviruses and 461 human papillomaviruses. And you see here phylogenetic trees. These are almost all or a large number of papillomaviruses here. And these are only the HPV, which split up in 5 different genera: alpha, beta, gamma, nu, and mu. And you see this here, that the precursor of all papillomaviruses split up in these 5 different genera. These phylogenetic trees, which measure evolutionary relationships, are based on the sequence of the L1 gene that you see here, which is the major capsid, which encodes the major capsid protein of papillomaviruses. So you see here the genome of HPVs, and this is a covalently closed double-stranded DNA genome with a number of open reading frames. Papillomaviruses are transmitted by direct contact from person to person that can either be close skin contact or sexual activities, and they can also be transmitted indirectly through contaminated objects and surfaces. The viruses are very stable. Work that has been done mainly on the mouse papillomavirus indicates that it survives 8 weeks on surfaces when it's on its own, free from a cell, and inside the squames that are the shedded keratinocytes, that it resides infectious for almost a year. Papillomaviruses replicate exclusively in keratinocytes. That's the main cell of mucosal and cutaneous epithelia. The other thing that's important, HPV are strictly host-specific. There are some exceptions among the animal papillomaviruses, for example, bovine papillomavirus 1 and 4. The natural host is the cow, as the name says, but they are also able to infect horses and cause sarcomas and carcinomas there. And then there is cottontail rabbit papillomavirus, that its natural host is cottontail papilloma. It's the cottontail rabbit, but it is also able to infect the pet rabbit and cause cancers there. HPV infections are locally restricted lesions. There is no viremia, so it's senseless to send in blood and test for papillomaviruses. Lucky for all of us is papillomavirus infections or lesions have a very high spontaneous regression rate. Let's talk about the clinical features. HPV can cause skin warts. These are common warts or verruca vulgaris, which are exophytic papules on many skin surfaces, mainly on dorsal hands and fingers. And in 3/4 of the cases, these are caused by 3 different HPV types, 2, 27, and 57. They occur very frequently in older children and in young adults. Then we have plantar and palmar warts, which are endophytic papules, and 90% of the cases are made of HPV 1, 2, 27, and 57. These are also very frequent in older children and young adults. And then we have flat warts, verruca plana, which are slightly elevated smooth papules, often at the upper extremities and the face, and they are mainly caused by HPV 3 and HPV 10. Studies have shown that skin warts, that 65% of the skin warts regress within 2 years, and at 4 years, already 80% of them have regressed, indicating that the immune system can get rid of them. Let me show you a couple of pictures. This is a histological picture. You see the wart, you see increased keratinization in the upper parts, you see the papillomas. Here going into the epithelium. You see that differentiation is slightly has changed. And here are some slides of skin warts on the hand, on the finger here, and again here on the finger. Then we have plantar warts that you can see here, here on the foot, and I'm not sure what this is. I think it's also a foot. And then we have skin warts on the hands. These are have been observed with people who deal with fishes or that work in chicken factories. It's not quite clear why they arise more often in these professions. And then what is important to note is that skin warts have a much higher incidence in immunosuppressed patients. And you see here skin warts under different conditions. This is a liver transplant recipient that receives immunosuppression. That's a renal transplant recipient also receiving immunosuppression. This is an CD4 lymphopenia, which means that that the patient has a genetic disorder, forcing him to produce less CD4 lymphocytes. This is a cancer patient who was treated with chemotherapy, which hurts the immune cells, and this is an HIV patient. This strongly indicates that that the papillomaviruses are targeted by the immune system, mainly by the cellular components, the CD8 positive T cells. In line with this, there are certain genetic disorders which have been shown to have a high incidence for papillomavirus infections. I think the most famous one is epidermal dysplasia verruciformis. It's a very rare autosomal recessive disease. To my knowledge, in the literature, there have been around 500 cases reported. And we know now that there are 3 different genes that are mutated in these patients. EVA1, also called TMC6, EVA2, which is also called TMC8, and CYP1. These patients have extensive infections, and in the wards you find mainly HPV from the genus Beta. And we have others provided evidence recently that the disease is most likely due to a specific immune deficiency that's not really understood how this is caused. These disseminated plain warts develop in early childhood and persist throughout life. And in sun-exposed areas, these warts can turn into a squamous cell cancer, and this occurs before the age of 40, almost exclusively on sun-exposed sites, indicating that UV light is an important cofactor for carcinogenesis. The other genetic disease that was discovered a couple of years ago by BZI and colleagues, it's called Treeman syndrome. It's very rare. There are only 4 cases in the literature. It's a generalized severe hyperkeratotic cutaneous papillomatosis. In 3 of the 4 cases was found HPV2, and the geneticists determined that it's an autosomal recessive CD28 deficiency. And the model, how this works out, is shown here. CD28 is a very important co-receptor for activated T cells after recognition with the T cell receptor. And this deletion seems to specifically lead to an increase in severe common warts or in giant cutaneous horns, which make up the Treeman syndrome. So it's believed because of the CD28 defect that the HPV infections cannot be cleared at all, but persist for a long time, which result in these giant hordes on the feet and on the hands of the infected patients. Let's go to the clinically even more relevant lesions that HPV can cause, and we start with anogenital warts. You see here different examples on the genitals, on the anus, penis for male that have all these genital warts here that you can see. Along these lines, I would like to introduce to you a very nasty disease called recurrent respiratory papillomatosis, and you see the warts or the papillomas here. It's also a very rare disease. Luckily, we have two different onsets, even at a younger age than it's in juveniles. The frequency here is around 4.5 per 100,000 children, and when it occurs in adults, it's only 2 in 100,000 adults. These are exophytic papillomas affecting the mucosa of the upper aerodigestive tract with a strong predilection for the larynx, as you can see here. And there are only two types mainly found, and this is HPV-11 and HPV-6. What makes these lesions so nasty and difficult is because these lesions are surgically removed, but they grow back, and this can occur very quickly in severe cases. And we have, I remember one case in Tübingen, he required 1 to 2 surgeries per month, which you can imagine is very, very difficult for these young persons. And it's very difficult for their quality of life. And I will tell you in the end, luckily, that when I went to the papillomavirus meeting a couple of weeks ago, that there is no therapy for this, at least in the US. Let's go to the most severe diseases that HPVs can cause, that is precancer and cancer of the anogenital tract. You can see the lesions here and here, and this is a histological analysis, and you see that this is that the cancer cells go through the basal membrane, indicating early invasion, that this carcinoma in situ has turned into an invasive, or will turn into an invasive cancer. Let me briefly show where this happens. Cancers are derived from the ectocervix here, or from the endocervix. And the main initiation site of cervical cancers, which is currently believed, is the so-called transformation zone, which distinguishes the single layer of columnar epithelia, shown here, from the squamous epithelia right there. These cells seem to be especially sensitive for HPV infections and turning into malignant cells. This slide here shows you that these infections progress over different stages, which are called LSIL, low-grade lesions, or in histology that's called CAN1, cervical intraepithelial dysplasias, grades 1, 2, and 3. And you see that the tissue architecture changes from very well differentiated to almost undifferentiated. And the more it progresses in situ carcinomas and invasive carcinomas, these cells look almost all like basal cells. The good thing for us is that this progress takes a long time. So after the initial HPV infection, it has to become persistent, and I will show you this in a couple of slides. The important fact from this slide is that it takes up to 10 years after the initial HPV infection that the precancer is formed, and it takes even longer until a full-blown invasive cervical cancer can be found. So there's a lot of time for interference. And it also tells you that the infection alone is not sufficient to cause cervical cancer, because these huge amount of time that is required. HPV infections cause more than 600,000 cancers per year. It's split up for the different cancers. So the main cancer that's caused by HPVs is cervix uteri, which I've shown you before, with more than 500,000 cases per year. And then we have cancers of the anus, vulva, vagina, the penis. And what has become more recognized in the past 10 years are the cancers of the oropharynx. And you can see here in this figure from 2022 that the oropharyngeal cancers now take over cervical cancers in the US. There is no good explanation for this, but this has also been seen in other countries, Great Britain, for example, but it's really not understood why these cancers become more prevalent right now. I've shown you in the beginning that there are many, many, many human papillomaviruses, and very early on it became clear that in the cancers in the anogenital tract, only a small subset of these viruses can be found. And you see here this. This is very old. This must be from the early 80s, mid 80s, and you can see here that the lesions are according to the disease grade are shown and the prevalence of the different viruses. And you see that HPV 6 and 11 and HPV, which numbers in the 40s, have a high prevalence in these low-grade lesions, but they are almost completely absent in invasive cancer, and their frequency is largely reduced in high-grade lesions. And then we have a couple of viruses, for example, 31, 33, and so on, that are very prevalent in the high-grade lesions, and their prevalence slightly decreases in invasive cancers. And then we have a couple of viruses. Right now, I would argue that's mainly 16, 18, and 45 that are incredibly prevalent in invasive cancer, like 16. And this has led to the epidemiological risk classification of HPV infections in the cervix. And I told you this in the very beginning that already 30 years ago, the WHO and especially the International Agency for Research of Cancer has, based on epidemiological evidence and molecular biology, has concluded that HPV 16 and 18 are carcinogenic for humans. And this has been expanded and 10 years later, and it's now clear that these types are regarded as class 1 carcinogens to humans. And based on this epidemiological risk classification, these types here shown in red have a high, when you are infected with them, you have a high risk of progression to cancer. So these viruses are called high-risk HPVs. And of course, in contrast, all the ones that have a low risk of progression to cancer, such as 611 and all these other ones, are called low-risk HPVs. And I'll show you this in a slightly different manner so that you can appreciate how bad HPV 16 really is. Let's first go to the right side. HPV 16 makes up more than 50% of all the HPV-induced cancers. And the second guy is HPV 18, and the other ones are much lower. And this slide, I think, highlights it even better because it not only shows the frequency here on the y-axis in the cervical cancers, and again you see that 16 is with roughly 60% and the worst virus we have out there, but you also see on the x-axis the prevalence in the population, and you see that there must be a clear difference between HPV 16 and HPV 52 because they are equally prevalent in the population when you're screened for them, but the fraction of positive cancers is at least 20-fold. There's a 20-fold difference between HPV 16 and 52, indicating that 16 has carcinogenic features than the other viruses do not have, which we still do not fully understand, I have to admit, unfortunately. I've mentioned this briefly. Sorry. You see here the prevalence of the infections, and you see that it rises in teens when they start sexual activity, and it peaks in the early 20s of the population, and then these prevalences go down again. When you look now, when the high-grade lesions occur first, you see that it peaks around the age of 30, almost 10 years later than the initial infection. And then when you look at cancer, it peaks around here. Also, there are many decades that need to pass before the cancer occurs. The other thing that is very important to understand for HPV causing cancer is that the majority of the infections are cleared by the immune system. You can see this here, that the number of cleared infection rises to almost 85% over time, and only a small fraction or a minor fraction of the infected women develop persistent infections. And these are the ones that will progress. So persistence is absolutely required of high-risk HPV for the development of cervical cancer. And this shows you that again that HPV-16 is the worst virus out there. It has the highest risk of progression over time compared to the other high-risk viruses in the alpha-9 group, where 16 and the other ones belong to. And this is also shown here for the alpha-7 group. Oh, that somehow it misses a couple of letters. And you see here in red that should be HPV-18, which has also the highest risk of progression over time, indicating that there's significant differences in these viruses between 16 and the rest and 18 and the rest. How do these viruses look? I haven't told you. So they are naked viruses, which means they have no lipid envelope. That's an icosahedric capsid that consists of 72 of these capsomeric structures, and they are composed of 5 molecules of the L1 capsid protein shown here that's used for phylogeny. And then there is most likely one L1 protein per pentamer, but it's not really clear whether every pentamer has an L2 molecule or only a couple of them. The structure is shown here again. So is an L1 pentamer, and then there's an L2 monomer, and inside is the double-stranded, covalently closed DNA, and it is packaged in histones, and it is very similar to the chromatin-like structures that our human chromosomes have. And this is quite unusual. Only small DNA viruses like papillomaviruses and polyomaviruses have a chromatin-like structure throughout the whole life cycle. What has been realized very early on that papillomaviruses replicate only in differentiating epithelium. And this shows you staining of biopsy material for 2 late proteins, the capsid protein L1 that I introduced to you already, and the other one is a non-structural protein that's called E4 that shows up in the upper layers of differentiating epithelium and is a marker for when the productive replication phase has started. And you see this for HPV-16, the high-risk viruses. You see this for low-risk viruses. You see this for skin wart viruses. The distribution is very similar. So the L1 and E4 only show up in the upper differentiated part of the epithelium. And it's shown here again. So that there's a specific histological structure in papillomavirus infected cells are called coilocytes. These are these cells with this enlarged nucleus and an almost empty cytoplasm. We know now that these cells are the ones that produce a virus and make infectious progeny virions. Let me briefly introduce to you so this differentiation dependency of the virus makes the investigation in the lab really tough. And we and others have developed models to investigate the HPV replication cycle. And it is not very complicated, but it's difficult to do in the lab actually. So these viruses are small. I told you 8,000 base pairs. They can easily be cloned and passaged in regular plasmids. So this is a cloned HPV genome in some generic plasmid backbone. And we remove the plasmid backbone either by restriction digest and then recircularize the genomes in vitro to get a structure like this, or more recently we started to use recombination in E. coli to get these structures. Then we take these viral DNAs and transfect them into recipient cells. We use normal human keratinocytes. And when we do this, we can establish stable cell lines that maintain replicating virus. And it looks like this. So we have, when we maintain them in as monolayer cells, indicated here by these squares, these are the infected cells. And we need to combine them with so-called feeder cells. These are mouse fibroblasts. And when we maintain the cells in this state, I'm not sure whether you can see this here. This is a keratinocyte island, and these larger cells are the fibroblasts that are growing around. And when you analyze what happens with the virus in these cells, you will see that the viral genomes are maintained at a low copy number, around 100 extra chromosomal copies per cell. And when you measure transcription, you will only find that the early genes are transcribed. There's absolutely no evidence for late gene expression or the production of virus particles. So we believe that this reflects the infection of these basal keratinocytes in the basal layer of the epithelium, and it is a non-productive maintenance phase. Let me now walk you through the early replication cycle in these basal keratinocytes. The virus attaches most likely through micro wounds on the basement membrane, and it attaches to heparan sulfate structures, and then a conformational change of the L2 protein occurs, and this goes hand in hand with the cleavage by a cellular protease called furin, and this enables the transfer of the virion bound to this heparan sulfate receptor to a second receptor on the keratinocyte cell that is unfortunately still unknown. And when the second receptor on the keratinocyte is recognized, the uptake of the virus occurs by a receptor-mediated endocytosis procedure. And you see then it forms these early endosomes, takes a couple of hours, and this early endosome turns into a late endosome. This takes around 8 to 12 hours. And then the virus, interestingly, still in a membrane vesicle here, will be transported to the nucleus, and it enters the nucleus most likely in the mitosis as a membrane vesicle with L1 standing out. And these vesicles go to so-called nuclear domain 10 structures, and then viral gene transcription occurs. And this is a very slow process compared to other viruses. It takes up to 24 hours until you see first transcription going on. To understand why warts look like warts, you have to appreciate that the virus infects one cell in the basal layer, and then it has the ability to expand in these infected cells, and this requires the activity of 5 different viral proteins. You need to replicate the genomes that's regulated by viral proteins called E1, E2, and E82. And you also have to induce cell division, and the proteins required for these are called E6 and E7. And there's also a process called genome maintenance that is important in dividing cells, and this is regulated by E2, and then I will show you in a minute what this really means. And this is a transcription map of high-risk human papillomaviruses, and you see we have a limited number of promoters, and there's mainly one promoter upstream of the early region that drives the expression of one precursor transcript extending from here to the early polyadenylation signal, and then via different splicing, all the proteins that I mentioned and other ones will be produced. In the differentiating epithelium, the late promoter is turned on, and this serves also to make some early proteins, but later on, it is the promoter required for the expression of the capsid proteins L1 and L2. Papillomavirus have a small number of conserved proteins. These are E1, E2, and E82. They are derived from, they share a large domain together. Then we have E6 and E7. High-risk HBV, that's one of the differences that we know. Additionally express an E6 star protein that requires splicing within E6. And then we have the late proteins, E1, E4, that's the E4 protein I talked to you about a couple of minutes ago. Then we have E5, that's only expressed or present in papillomavirus from the alpha, mainly from the alpha genus in HPVs, and then we have the capsid proteins. So only a very small number of proteins that these viruses express. Let me first talk about HPV genome replication. Between the late and the early region is a region that's called upstream regulatory region that consists of transcriptional elements and replication elements. A replication element is the origin of replication that you see here that consists of multimerized binding sites for the viral E2 protein and the E1 protein. And we know now that E1 and E2 form a protein complex that has a very high affinity for the origin of replication. And after E1 is loaded onto the viral origin by E2, E1 forms a double trimer that then is turned into an hexameric structure of E1, and two of these hexamers form over the viral DNA. And this is the active replication enzyme that uses ATP to unwind and separate the strands of the virus. And E1 recruits cellular replication enzymes, polymerases, DNA polymerases, primases, replication protein A, topoisomerases, and so on, to finally achieve the replication of the viral genome. So the virus has no own polymerase or anything, only the E1 protein to mediate or to enable viral replication. The rest must be provided by the host cell. E2 not only is essential for genome replication by formation of the replication complex with E1, but it is also transcription factor. It can activate and repress viral gene transcription through the interaction with a cellular protein called BRD4. And E2 is also very important for this genome segregation function I mentioned briefly before. And when you remember, so when cells divide, they go through mitosis, and in mitosis, the nuclear membrane is dissolved and is reformed at the end of mitosis. And so the viruses need, obviously, to ensure to be in the nucleus again. So what E2 does, it binds via its DNA binding domain to the viral genome. And with its N-terminus, it binds to mitotic chromosomes so that the viral genomes end up where the chromosomes are, and this is in the newly formed nuclei. What we have also seen is that a highly conserved transcript among papillomaviruses, I have to say, is the E82 transcript, and it links the small open reading frame to the C-terminal half of E2, which is where the DNA binding domain and the dimerization domain of E2 proteins is. But it completely replaces the amino terminal domain of E2, which is important for the transcription activation, genome replication, and segregation function of E2. We and others have shown that the E82 protein is mainly a restriction. It restricts genome replication in undifferentiated cells, and this is highly conserved. So this was shown for different for high-risk viruses, for low-risk viruses, and for viruses from other genera. So this is extremely conserved. And we know now that this E82 has different activities to achieve this. First of all, it is a very potent repressor of the basal activity of all viral promoters that have been tested. It is also able to inhibit the E2 transactivation function, indicating that it can counteract E2 activity. And this is not only in the context of transcription, but also in the context of origin replication. We and others have shown that E82 directly inhibits E1, E2 dependent replication. And this all explains why the knockout of E82 in HPV genomes results in a higher replication than usual. Let's now come to the so-called oncoproteins. Very early on, it was clear that all cervical cancers and cell lines that were established from these cancers, such as HeLa, always express the E6 and E7 genes of high-risk HPVs, and these E6 and E7 are encoded here in these genes shown in red. Very early on, it became also clear that only the high-risk E6 and E7 proteins have oncogenic activities. And the assay that was performed looks like this. When you take normal human keratinocytes and you try to passage them in culture, these cells will divide a couple of times and then will start to senesce and no longer grow. That's usual for all human cells when you take them into culture. When you now make these cells transgenic and introduce HPV 16, E6, and E7, these cells will never stop to grow. There's a continuous growth, and so these cells are immobilized. When you now take E6 and E7 proteins or genes from a low-risk virus, like HPV6, these cells will essentially behave like the negative control, and these cells will not grow out, but will go rather in an irreversible growth arrest. And many studies have shown that this is a unique feature that all high-risk viruses have, and very few other HPV types have. So it's very distinctive. How is this caused? And to understand what drives continuous growth of cells, we need to understand the cell cycle. I'm pretty sure you know all this, but let me briefly make the major point here. It is very crucial for cells to control when they start DNA synthesis and finally when they want to divide. This is called the G1S restriction point or G1S checkpoint. It decides whether the cells go into S phase and start replicating the DNA. This restriction point is regulated by a protein family that's the retinoblastoma protein family. The most important member is the retinoblastoma protein, here abbreviated PRB. This protein has the ability to bind a class of DNA binding transcription factors called E2Fs. And normally, PRB is inactivated by phosphorylation by a class of kinases called the cyclin-dependent kinases that are regulated in a cell cycle-dependent manner. When RB is phosphorylated, it is released from the E2F factor, and E2F is now an active transcription factor and produces mRNAs. And these proteins drive the progression into S phase, and not only the progression into S phase is encoded by these proteins, there are also DNA replication proteins, DNA repair proteins, and so on. So basically, this is the key to drive cells into S phase. And what the high-risk or HPV E7 protein does, it binds to RB and can get it out of this E2F complex without the need for cyclin-dependent kinase phosphorylation. So even if the cells does not receive the signals which are done by growth factor signaling, E7 containing cells will disrupt this and will turn on S phase genes, and so the cell cycle will progress when E7 is present. However, cells are not stupid. Cells will sense this, and this unintentional, or it is intentional, this overactivation of the cell cycle causes DNA damage, and it behaves like an oncogene. And we know from zillions of studies that the p53 tumor suppressor protein is a stress sensor that detects such changes, and it responds with the regulation, with the transcriptional regulation of a large amount of different genes that can cause either cell cycle arrest, or the cells go into cell death called apoptosis. The cells can go into a permanent cell cycle arrest called senescence. They can also induce differentiation or DNA repair. So when you do this, when E7 alone is in a cell, you will observe increased p53 levels, indicating that p3 is activated and can do all these things. Arrest the cell cycle by upregulating cyclin-dependent kinase inhibitors, or induce pro-apoptotic factors and go into apoptosis. And what has been found that the second protein that is required to immortalize keratinocytes, the E6 protein, and this is exclusive for high-risk HPVs, will bind a ubiquitin ligase, an E3 ubiquitin ligase. That's a protein that will attach ubiquitins to target proteins, and this can lead to the degradation via the proteasome. So high-risk HPV E6 will form a trimeric complex of E6AP and P53, and this will lead to the specific degradation of P53, which means in E6, E7, in E6 positive cells, the levels of our P53 are very low. Normally, this is regulated by a cellular protein called MDM2, and this responds to diverse signals from the DNA damage signaling pathway. However, when E6 is around, this all does no longer matter. P53 is specifically degraded by this complex. So this means a cell that is infected by a high-risk virus and expresses E6 and E7, E7 will disrupt RB, leading to proliferation, and the signals that are generated, replication stress and the DNA damage response that activate P53, are blunted by the activity of the high-risk E6 protein because it degrades P53. When we come back to our model, how these cells expand in the basal layer, it becomes now clear that a genome replication is important to maintain a copy number, and the genome maintenance function of E2 is very important. But it is also very crucial for these cells to disrupt P53 and RB activity to expand. And disruption, I've tried to convince you what happens in the basal layer of the cell, but initially I showed you that the productive replication cycle only occurs in differentiated cells. And you have to understand that normal epithelium, the only cells that will divide are in the base layer. And these are division competent stem cells that replenish the epithelium. And these cells will divide horizontally and vertically, And when these cells enter the suprabasal layer, they will undergo a terminal differentiation program and resulting in the different layers of differentiating epithelium. What is important to understand that these cells exit the cell cycle. They are post-mitotic. They differentiate, but they are in a phase that's called G0 that is very similar to the G1 phase that are shown in the cell cycle. So these cells will not undergo DNA synthesis. I've shown you okay, let's go a step back. So to investigate the productive replication cycle, we need to differentiate the infected keratinocytes that are shown to you. To do this, we use so-called organotypic three-dimensional cultures, and you take these cells from the monolayer and you seed them onto a collagen plug with embedded fibroblast cells to mimic the dermis. And when you do this and seed the HPV-containing cells as a monolayer on these cells and expose them to air from above and feed them with medium from below, these cells will start differentiating. And you can see this here. This is immunofluorescence analysis with antibodies of these organotypic cultures. And you can detect by immunofluorescence that the late proteins E4 and L1 are made in the upper layers, and also that the viral genome amplification occurs only in the upper layers of the epithelium, indicating that this is a good way to analyze the late events of the replication cycle of high-risk HPVs. I wanted to show this. We took this picture. This is how these organotypic cultures look when they are grown. So we had, these are the medium exposed cultures. We do this. This is the collagen plug here, this whitish color. And we keep them with medium on top until they are confluent, and then we expose them to air, and then they look like this, and then they are fed from below. This whole process takes approximately 2 weeks to finish. People have done, we and others have done this extensively, and so we now know that there's this non-productive maintenance replication function I've showed to you, and then there's this productive replication going on in the suprabasal layer. Very early on, it became also clear that high-risk HPV, all HPVs, but mainly high-risk HPVs, change the behavior of the differentiated cells. And it turns out that in the suprabasal layers, these infected cells now express markers that are indicative that these cells are in the S phase, which means they have re-entered the cell cycle. And these are typical proteins that that the cell needs to replicate its own DNA. But I told you in the beginning, these are very small viruses. The only enzyme they have is E1, and its function is to unwind the viral DNA and recruit viral replication factors that would normally not be present when these cells are not in S phase. So you need to get the cells into S phase to get the virus replicated. And it's not only that they induce an S phase, but you can see in this experiment from my paper from Wang and colleagues that that DNA synthesis is induced in these differentiating cells. So these are these organotypic cultures. These are the normal keratinocytes, the uninfected ones up here, and then there's HPV-18 infected cells. And when you measure now the extent of DNA replication by the cell in the different layers, and this is done by adding bromodesoxyuridine, which can be incorporated into newly synthesized DNA and can be detected by a specific antibody, you can see that the uninfected cells, DNA synthesis only occurs in the basal layer, whereas in HPV-18 infected epithelium, many suprabasal cells start replicating the DNA. And when you specifically look at the viral DNA, it turns out that massive viral DNA replication only occurs in these differentiated layers. Of course, people try to understand which protein is responsible. And from what I've shown you from the monolayer basal-like keratinocytes, the answer is quite obvious. It's most likely E7. And this was proven here. So people generated an HPV16 genome that has an E7 knockout, and they looked at these at BRDU incorporation as a measurement for DNA synthesis, and they looked for DNA replication proteins, and you see that the E7 minus mutant has much lower levels in the suprabasal layer of all these markers than the wild type. Strongly supporting the idea that E7 also induces DNA synthesis in the suprabasal layer, and this is also mediated by the inactivation of RB. And again, P53 not only plays a role in the basal layer, but it's also important to achieve replication of the viral genome. So these in this paper, they generated an HPV, I think it was 18 E6 knockout genome, and you see that this is in red is the viral DNA replication. You see that the rate signal is basically gone, and P53 reappears. And they were able to complement this with E6 overexpression, and you see that P53 is gone, and viral DNA replication is induced again, indicating that the P53 degradation function is also important in suprabasal cells to allow viral DNA replication. So I've told you that these proteins are oncoproteins, but they have not evolved as oncoproteins to cause cancer. They have evolved to enable the expansion of virus-infected basal undifferentiated keratinocytes. And they have also evolved to allow virus replication in differentiated keratinocytes. So one should call them not only oncoproteins, but rather as essential replication proteins of the virus. So the interest in HPVs comes from the fact that they can cause cancer. When you look at cancer as a virologist and ask, are cancers productive? So is it good for the virus to induce a cancer? Is this a way to increase virion synthesis and production? The answer is this is not the case. So this is a staining of a low-grade lesion, a productive lesion. You see here the signals for E4 and L1, the capsid proteins. And when you now stain a high-grade lesion, that's a carcinoma in situ, for example, you see very reduced E4 expression, and you see very minor or basically no expression of L1. So we know now that these cancers are abortive infections and that virus is not produced in them. So the virus has no advantage of inducing a cancer. So the cancer induction is an unintentional consequence of E6 and E7 expression in persistent lesions. And I will show you what people believe right now, how high-risk HPV causes cancer. I think you heard me talking about it a lot, so I think you will know what comes now. Oh, no, maybe not, maybe not. So one thing, an important fact that I forgot to mention is very early on, it became clear that in cervical cancers, high-risk HPV genomes are not present in this extra chromosomal state, this plasmid state that is required for the generation of progeny virions. But very often the viruses are integrated. 70 to 80% of the cervical cancers, you find the viral genome integrated into the host DNA like this or multiple copies like this. And what has now become clear through all these huge next generation sequencing projects is that integration occurs integration occurs in a way that E6 and E7 expression is retained, whereas the expression of other viral proteins is mostly absent. And I think that this leads to an uncontrolled E6 and E7 expression that can no longer be controlled by the virus. These integrated genomes can no longer induce late viral proteins. They cannot amplify the genomes so that the whole late phase can no longer be achieved. The other thing that has been observed is this insertion into the host chromosome can lead to an insertional mutagenesis, which means that flanking genes can be activated by the virus. And integration by itself can cause can contribute in addition to the activities of E6 and E7 to cancer progression. So currently, what we believe is the following. The long-term expression, I told you, for cervical cancer to occur, the viruses have to become persistent, which means you have infected cells that continuously express E6 and E7. And because E6 and E7 override the cell cycle checkpoints RB and P53, mutations can no longer be repaired as efficiently as they should be. And it is known from observing these cells over a long time that there is a lot of genomic instability going on when cells express high-risk E6 and E7, increasing the mutation rate in these cells. What has also become clear that the genomes not only accidentally integrate at genomic loci, but this is a very fluid situation. People see now, based on NGS sequencing, that recombination can occur, and that flanking parts can be amplified and rearranged. Sometimes these parts with the virus can be excised and reintegrate. So having a high-risk virus in a cell generates a lot of genomic instability. So we think now that the combined activities of E6 and E7 and these unstable extrachromosomal genomes result in all these additional mutations, of some of which can drive cancer progression. And this also explains the long time it takes until cervical cancer after the initial infection how long it takes until cervical cancer really occurs in infected people. So let me briefly talk about therapy. And this is, I think that's one of the sad stories of papillomavirus research. Basically, with skin warts and genital warts, you can freeze them and cut them out. You can treat them with 80 to 90% trichloroacetic acid. You just denature them and cut them away. There's surgical removal. You can do laser therapy. You can prepare ointments with all these substances and put them on skin warts and genital warts, and very often they will disappear. But this is not very specific though for the virus. It's even worse with pre-cancer and carcinoma and cyto and invasive cancer. These things do not work or cannot be done. And there's only the chance of surgical removal, and people are then treated with radiation therapy and a combination of radiation therapy and chemotherapy. And this is, to my knowledge, still cisplatin. As with every cancer, the outcome depends upon tumor size. And if metastasis has occurred into the lymph nodes, and the 5-year survival rates for cervical cancer patients can be very good, or 85% if it's late and the cancer has spread, it's only 7%. So still, we know this now for more than 30 years, that it's E6 and E7 and so on. We still do not have specific antiviral therapy. The only good news that I have for you is RRP, recurrent respiratory papillomatosis caused by HPV 6 and 11. The FDA approved in 3 months ago in August 2025 an immunotherapy where certain early antigens are overexpressed with an adenovirus, and this results in the regression of these warts. But for high-risk viruses, we have nothing. So the best, and this is old news, the best way to prevent high-risk HPV-induced cancers is to prevent high-risk HPV infections. And so people in the 90s decided they should develop a vaccine against HPV. And they made use of the fact that they realized when they only expressed the major capsid protein L1 in either yeast or in insect cells, the recombinantly expressed L1 protein will form these pentameric structures, and these molecules will also form capsid-like structures that very much resemble native virus particles, and they were called virus-like particles. There are now 3 to 4 different vaccines on the markets. I think the so-called Gardasil 4, which targeted 2 low-risk viruses and 2 high-risk viruses, is now completely replaced with Gardasil 9, which includes 9 different HPV types, 7 high-risk types, and 2 low-risk types. Cervarix was targeted HPV 16 and 18 only, and there is now, I think it's very new, Chinese vaccine that also targets HPV 16 and 18. You can see here the doses of L1 protein. These vaccines are produced in insect cells, in yeast cells, or in E. coli, and most of them have to be injected 3 times with these schedules shown here. And they are licensed to many countries. The Seculin, I'm not sure whether that's only in China or whether they try to expand it. What has been shown in a large number of studies that all these vaccines have the capability to very efficiently prevent persistent infections and precancer formation. You can see here the different studies. There were 10,000s of participants in these studies, and they work against CIN, they work against vaginal intraepithelial neoplasias, they work against genital warts, they work against persistent infections, they work against anal infections, and so on. And I also read that in Australia, they were the ones with the first population-wide vaccination programs, that also the incidence of recurrent respiratory papillomatosis is going down, indicating that it also works against RRP. Some people doubted whether prevention of high-risk HPV infection would really reduce cervical cancer, which is, from my point of view as a virologist, really not a very good argument. But the data are out there now from 3 different European countries, Sweden, Denmark, and Scotland. And you see in red the unvaccinated women. And the best thing is to compare it with girls or young women vaccinated at an age below 16 or 17, or here below 12 and 13. And you see that the red line that you get an increase in cancer development in the unvaccinated population, where in the vaccinated population, you basically see no more cancers, strongly suggesting that HPV vaccination really, really prevents cervical cancer. Just about Gardasil 9. So they also included 5 more high-risk types, 31, 33, 45, 52, and 50. They again made a large trial, more than 14,000 women. And basically the conclusion was that they saw a 96.3% reduction in high-grade disease for the other 5 types, not included in Gardasil 9, in Gardasil 4, sorry. And again, so the whole story of Gardasil 9 is that it is as efficient as Gardasil 4, but is more efficient against the 5 other types, and it has no disadvantages. So what is the mechanism? It has been shown that these virus-like particles induce long-lasting high titers of neutralizing serum antibodies. You can see this here. So people have been followed over 11 years now. Whereas the natural immunity in terms of antibodies against HPV is very, very low.
Okay.
Excuse me? Okay. Whereas the natural immunity is very low when you vaccinate with the normal regimen of 3 doses, you get long-lasting high titers of anti-L1 antibodies. And you can test whether these have neutralizing capability. And because it is so difficult to make authentic HPV virions, people have come up with the idea, oh no, because of the vaccine development. Okay, something that I should know? Oh, so authentic HPV virions are very difficult to make. You can make them from these organotypic cultures, very inefficient. But what people have realized is when you express the 2 capsid proteins, L1 and L2, they will form capsids, and they will package basically any plasmid DNA that you introduce together with them in cells. And you can make, you can introduce, or you can package reporter genes like luciferase or other reporter genes. And this assay here shows so you infect cells, and then you can add antibodies either from vaccinated people or wherever you have them from. And you can see when you add no antibody, you get activity of the reporter. When you add now a highly neutralizing antibody like this V5 antibody, you no longer get activity. Whereas when you take an antibody that has no neutralizing activity, you get high activity. So you can test with these systems neutralizing activity of the antibodies, and it was shown that the vaccines do this quite well. And I think one of the more elegant experiments that I've seen is that I think it was John Schiller's group at the NIH have shown this in an in vivo assay. So they used mice that were vaccinated, and then they took serum and diluted it, and they transferred this immune serum into naive mice, and they then infected these mice intravaginally with this HPV pseudovirus that expressed in luciferase, and then did in vivo imaging. And you can see here the mouse that has received the pre-immune serum before vaccination, so without L1 antibodies, and then being infected with this luciferase reporter. And you see that there's a strong luciferase signal in the vaginal tract, whereas when the mouse was treated with this VLP serum, this luciferase signal does not occur, strongly indicating that there was no infection because of the antibodies. So what is the mechanism? I've shown you this before, and it turns out even low levels of antibody are sufficient to prevent infection, and it is believed that low levels of antibody do not necessarily prevent the attachment to the heparan sulfide, the first receptor, but they prevent a transfer to the second receptor on the keratinocyte, so preventing infection of the keratinocyte. And the other mechanism is when you have high level of antibodies, of course, the virus will be covered by antibodies, and these antibodies will cover relevant interaction sites with the first receptor, so the virus can no longer bind to the first receptor, and then it will be recognized by immune cells and will be depleted it will be depleted. So why are these antibodies so effective at preventing infection in vivo? And the idea that is out there that there is not one neutralizing epitope that is on these VLPs, but people have shown that there are multiple distinct L1 epitopes, and each epitope can be targeted by a different antibody species, by a different antibody. So these VLPs induce an immune response that is very effective preventing infections with even HPV-16 variants. And one thing that I have not mentioned, as these are DNA viruses, so they mutate very slowly. It's not like HIV or SARS-CoV-2 or any of these viruses. So, and because they evolve so slowly, it's very unlikely that you get in a brief time a large number of escape mutants. And because these antibodies target distinct epitopes on L1, this is also very unlikely that a virus occurs that can mutate all the epitopes and still be functional. So I think we're safe for a long time. So let me come to my final point. Screening. So HPV and cervical cancer screening is, of course, done. I know this mainly from the US and Europe, I have to admit, has been done for a long time. And this can be very efficient when done properly. And the reason for this is the lesions, I told you this, the lesions take a very long time to progress into invasive cancer. So if you find precursors, there is time to intervene with it by removing it surgically. You need only to detect these lesions early. And there are basically two concepts that are not necessarily mutually exclusive. It has been done for a long time, a screening for precursor lesions in cervical swabs, and this is done by cytology. And then in the past, I don't know how many years, it has become obvious that screening directly for the virus in cervical swabs, when you look for DNA or RNA by a zillion of different diagnostic tests that are out there, that might be even better. And just to explain cytology to you, and I'm not a cytologist, I'm not a medical doctor at all, so I have very basic knowledge about this. But what people do is they use the cervical swab and they streak it out on a microscopic slide, and they do an H&E stain, and then people are looking at it and trying to understand whether they have abnormal cells that are indicative of high-grade lesions looking at this. And I was told, because it is a rare event, it can be very difficult to detect this, especially in the borders. But people do this, and they have been doing this very successfully, so it seems to work. And I know that this has now been automized, and I think that artificial intelligence is coming into play and making the detection even better. And I'm sorry for this. These are my last two slides, and they are in German because I received them from Professor Effner and I could not modify them because they are graphs, and so sorry. But I think you can get the point. So in Germany, screening for women up to the age of 34 is done by cytology on a yearly basis. And when some high-grade lesion is detected, they are transferred to a gynecologist, and he will do colposcopy, which means he will look on the cervix and see whether there's an abnormal lesion that needs to be removed. And if these low-grade lesions are detected, and depending on the age of the patient, then an HPV test is included. And when this is positive, the woman goes into that direction. And so she's referred to the gynecologist and is doing colposcopy. If it's HPV negative, then she can go back. In older women, which means at above 35, there's a combined cytology and HPV test every 3 years, and depending on the outcome, let's look here, it's negative. The HPV test is negative, but the cytology tells you there might be something going on. So then they wait for a year, and if it's then HPV positive, she's referred to colposcopy. If it's negative, she goes into the screening again. If she's HPV positive and has a high-grade lesion, then she goes immediately into colposcopy, and so on and so on. I apologize for this, but I really know this only for Germany and not for other countries. I'm very sorry. And with this, I would like to end, and I'm open to question. And now I need to figure out how to see you guys, because there you are. There you are. So this is it for me. I'm happy to take questions if there are any.
आप में से किसी कोई क्वेश्चन है? लाउड स्पीकर है? लाउड स्पीकर कहाँ है?
Oh, the sound is not very good. Is my sound also bad? No. Oh.
Are you listening to us? Mike, man, कहाँ है लिखा हुआ है? ये? So come here, please. Can you hear me?
Khalid, you talking to me?
Yeah.
I can hear you.
Okay. So, any question from the audience? Yes, please.
Oh, you didn't hear me? No, you heard my talk, or?
No. There was a bit problem within 2 slides, so we resolved it, and that was okay.
Okay. Oh, I'm sorry.
Oh, no problem. So, we have a question from the audience. Yes, please.
Hello, can you hear me?
Yes, I can hear you.
Okay, sir, my name is Navira, and my question is that how we can differentiate that between the plantar warts, or can they appear on fingers as well,
Oh,
or are they serious, or like
no.
they go by their time?
No, the plantar warts are not serious. They do never progress into cancer. Most of them will go away until over a couple of years, until you have some genetic background, and they can be removed. But I'm not a dermatologist. I'm sorry. I'm not very good at treating people. I've never done this.
Okay. Thank you so much.
Any other? I have some questions regarding, with the vaccination. So we have pentavalent vaccine, non-valent vaccines, just like we are expressing the L1 protein in different vectors or different cell lines, and then later on combining, or then we are all expressing in a single system, and then how we can do this for the vaccines, for VLPs.
So you want to make them by yourselves? No.
I want to try it.
Oh, well, okay. But you know that I'm not so Why don't you
Means
use these other the vaccines that are around? So what about the Chinese vaccine? I heard at the meeting that it's basically very similar to the vaccines from Merck and GSK. Is that available in Pakistan?
yeah, we recently have started the vaccination for HPV recently for the first time. But just I should know how to synthesize our vaccine
Oh,
is. So there is a lot of the
so
space with projects in this direction.
what I can tell you is that people started initially in the 90s trying E. coli for expression, and they all failed, and then they switched to yeast or to the insect cell system. But now the Chinese have come up with a way how to do this in E. coli. So maybe you should read I'm not sure what has been published, how well they describe how they do it, but I would go
Oh,
for their protocol because I heard from all the guys that have invented the initial vaccines that the data look very, very good for the Chinese vaccine. So it seems to be as efficient as the insect or the yeast produced vaccines.
okay. The second question is my regarding the essay which I was a long time been working with you. So our CH cells, I just want some questions like CH cells. We know that they are expressing E6, 7 like this. So we should need to transfect them with the E6 or E7 or the indigenous E6 or E7 is enough to degrade P53.
Yeah. So
Yeah.
what's what's the question?
The question.
How to interfere with it? No,
Yeah, no.
no.
Actually, I want to ask that CH cells are expressing indigenously the E6 and E7 proteins
Yeah,
of HPV. Yeah.
yeah.
So if I want for the drug screening and if I want to see their interaction with the P53, I should transfect the CH cells again with the E6 or E7 or the indigenous is enough to degrade the P53.
Oh, so there's sufficient E6 in there to degrade P53. You do not need more. We only
Oh,
did this big as a simple system, but
Okay. Just I have to transfect with the P53 and then I should see that it is degrading or not. Yeah.
this should work. Yeah, yeah.
Yeah.
So
So
we use now very often this. So we have this fusion protein between P53 and the luciferase, and you can use a luciferase. No, no.
okay. Yeah. So the other question is that can I validate by the transfection of E2 protein this process in CH cells that can I inhibit the
Yeah.
E6?
That has been
Yeah.
published long time
Oh,
ago. That you can either use a E2 or E82, and they will inhibit expression from the early promoter, and E6 and E7 are gone, and the cells will go into senescence, and some will die because of apoptosis.
okay. Thank you very much. If anyone has any question, yes, you can come, please. Yeah.
Hello, Professor. Can you hear me?
Hello? Yes, I can.
Thank you, sir. I get a lot of knowledge from your lectures. So I have two questions. One question is why is human papillomavirus more infection in uh, females as compared to males? Secondly, why it don't have antiviral drugs, specific
I,
antiviral drug, antiviral?
I'm not sure whether women are more infected than men,
Yes.
but the lesions are very obvious. Only women have a cervix, and this is where the cancer arises. So they have been tested more often than men, and it's much easier to test them. Uh, people have tried to do swabs from the penis, but it is not very reproducible for some reason that I do not understand. Maybe because, there's not a lot of mucus around. Maybe it is hard to get the right cells with virus. So, but it's sexually transmitted. So, men must be infected, otherwise the system would not work.
Secondly, Professor, why we don't farm specific antiviral drug for this virus?
I have no clue. I know that many people have tried, and at the most recent meeting, a, a, a a colleague in the US, he has claimed that they have developed an anti-E6 small drug. And in the lab experiments, it looked very, very good. So, but
Okay.
now they will go into, humans and will try to figure out whether it really works.
Okay, any other question from the audience? Okay. Okay, if there is no question, we should wind. I am very thankful for this
Yeah.
much informative lecture, and we will be in contact with you.
Well, okay. Thank you for the invitation, Khalid.
Thank you, sir.
Thanks a
Thank you
lot.
for your lecture.
Okay. See ya. Hear from you.
See.
Bye.
Bye. काट दिया छोड़ दिया हाँ,